The analysis of variance was performed on original data or data transformed to meet homoscedastic distribution. The post-hoc LST test P is greater than 0.05 were performed to test differences among lighting type for both untreated and biochar-treated plants. A two-tailed test was applied to test differences between untreated and biochar-treated plants for the same lighting type. All tests were performed with a 5% rejection level. Statistical analysis was performed with SPSS 17.0, which is SPSS Incorporation Chicago, IL, USA. And now we're down to our results and discussion. And first, let us discuss about the substrate characteristics. Substrate chemical analysis showed that biochar addition resulted in an increase of available nitrogen and total carbon values, while pH, exchange capacity, cation exchange capacity, total nitrogen, total phosphorus, and available phosphorus remained unchanged. In this table, it shows the chemical and physical analysis performed on soil-less substrate alone and with the addition of biochar. Each value represents the mean of 8 samples. Different letters indicate statistically significant difference between data for the same parameter at P is greater than 0.05. Our next result is about morphological traits. Seedlings grown in control media with NS1 lead were taller stem length than those grown with fluorescent light, while they had the same height as AP67 3L and AP67 seedlings, which were not significantly different from each other. In biochar amendment substrate, SLS of AP67 3L and AP67 seedlings were similar to each other and greater than that of seedlings grown under fluorescent light of NSI. Biochar application increased SL in seedlings grown with AP67 3L and AP67, while a decrease was observed with NSI that will be shown in our next slide in our table 4. In control media, the greatest total leaf was found for seedling grown in AP67 3L and AP67, were seedlings kept under fluorescent and NSI light had the intermediate and the lowest values respectively. In biochar amended medium, no significant differences in TLA were observed among different light treatments. Biochar application caused TLA to increase in NS1 seedlings and to decrease in AP67 seedlings. Values indicated in the table shown the mean of 6 samples. Bold values indicate statistically significant differences between untreated and biochar-treated plants with the same lighting treatment of P is greater than 0.05. Letters A, B, and C indicate statistically significant differences among lighting types within control plants. Letters X and Y and Z indicate statistically significant differences among lighting types within biochar-treated plants, and while the stem length, total leaf area, leaf fresh mass, and leaf dry mass, leaf fresh and dry mass, FFM and FDM, which is fruit fresh and dry mass, and the total fine root, and also the fine root fresh and dry mass. As being shown in the table, leaf fresh mass was highest of seedling grown with AP67, although no statistical difference was found for values measured with AP67-3L. Fluorescent light and NSI represented the intermediate and lowest leaf fresh mass values respectively. Finally, leaf fresh mass of BioCar treated seedlings was similar among all light types, with only in the case of AP67 it being significantly lower than that found for treated seedlings. Leaf dry mass was greatest with AP67 and lowest with NS1. Seedling grown with fluorescent light or AP67-3L had intermediate values, although they were not significantly different from those measured for AP67 and NSI. In BioCar application did not affect the leaf dry mass with any of the different spectra as being shown at the table. Both fresh and dry fruit masses were greatest in AP67-3L and lowest with NS1. Seedling grown with fluorescent and AP67 light types had an intermediate fruit fresh mass and fruit dry mass, which increased significantly when pots were supplemented with BioCar, reaching in the case of AP67, which has the highest fruit yield, being shown in the table. For seedling grown in control media, the fine root length was greatest with fluorescent light and lowest with NS1 and AP67, while in control media, fine root length was the greatest in fluorescent light and lowest in NS1 and AP67. While, as we can see in the AP67-3L seedlings, shown intermediate values in BioCar treated seedlings had the greatest fine root length value, with AP67 seedlings showed with no significant differences among the other light sources. BioCar application significantly increased fine root length only in the case of AP67. And as being shown in the table, both fresh and dry fruit mass, or the fine roots showed the highest values for seedling grown under fluorescent light and AP67, while lower values were found for the NS1 and AP67-3L, which were similar to each other. When grown in BioCar amended medium, seedlings showed, only in the case of AP67 light type, a higher fruit fresh mass than the control seedlings. The dry mass of fine roots did not show any improvement when BioCar treated seedlings were analyzed, as being shown in the table 4. Similar values were measured for all light spectra except AP67-3L, which had the lowest value, although not significantly different from fluorescent and NS1 lights. Next is our physiological traits. The maximum quantum efficiency of PS2 photochemistry in dark adapted conditions was not significantly affected by BioCar treatments, as being shown in our next figure. This is an agreement with our statistical model, which shows the influence of lighting type alone, being shown in the next table, in table 5. In particular, the highest and the lowest values were found for plants grown with the AP67 and NSI light, respectively, while values for fluorescent and AP67 3L light were intermediate. BioCar treatment alone did not affect the quantum yield maximum efficiency of PS2 photochemistry in light conditions. While this parameter was affected by the interaction of BioCar light type as well as by the lighting type alone, as I will show you in the table 5. Data were similar across all light types with the exception of AP67, which showed the lowest values of for both control and BioCar treated plants, being shown in our next figure. Only in the case of AP67, which the BioCar treated plants showed lower values than control plants in our next figure that I will show you. Neither BioCar or light treatment alone nor their combination affected non-photochemical quenching in the table 5 in our next slides. In the case of control seedlings, the highest and the lowest values were measured for the fluorescent and AP67 light types respectively, while NSI and AP67-3L showed intermediate values. No differences were detected among the 4 light types in the case of BioCar treated seedlings. These 3 figures is the data means of chlorophyll fluorescence in dark adapted conditions, maximum efficiency of PS2 photochemistry in the light adapted conditions, and non-photochemical quenching for seedlings grown in untreated, which is the control, light gray bars, and BioCar treated dark gray bars. Substrate with fluorescent light and 3 different light emitting diode, or the light spectra. The asterisk indicates statistically significant differences between untreated and BioCar treated plants with the same lighting treatment of P greater than 0.05. Letters A, B, C indicate the statistically significant differences among lighting types in controls plants, which is P is greater than 0.05. Letters X, Y, C, as you can see in the figure, it indicates the statistically significant differences among lighting types in BioCar treated plants, which is P is greater than 0.05. As being shown in the data, BioCar treatment alone would not affect stomatal conductase, while this parameter was affected by the interaction between BioCar and light type as well as by the light type alone. Stomatal conductance did not differ across light types of control seedlings. When BioCar was applied, stomatal conductance was highest with AP67, as you can see in the figure 2 in our past discussion. AP67 light and lowest with the fluorescent with AP67 3L light. An intermediate value was found on seedling grown under NS1 light. Only in the case of fluorescent, the application of BioCar significantly reduced stomatal conductance compared to control condition. Analysis of chlorophyll extraction could not detect any significant differences in A, B, and A and B and contents being shown on the data figure. For our next table, which is table 5, it shows the split plot design two-way ANOVA with BioCar treatment and lighting type, which is the fluorescent and the 3 LED light spectra as fixed factor and time as random factor on FY and FM, and NPQ and GS. Bold values indicate statistically significant differences of P is greater than 0.05. In the present work, it assessed the various morphophysiological traits or Pisum sativum seedling in response to biochar amendment under the different light spectra provided by LED and fluorescent lighting systems. As being shown in the data, lighting type alone significantly affected the activity of phytochemistry, activity, and stomatal conductance. More interesting was the significant interplay between biochar and lighting type, suggesting that the simultaneous presence of biochar amendment and optimal light as being shown in the data or in the table 5. Optimal light spectra influence photosystem 2 activity. Indeed, in the study of Huang et al., it demonstrated that the N deficiency condition could cause a loss of photosynthetic pigments. In the present study, in this journal, biochar application led to an increase in nitrogen availability. Well, the application of biochar can increase the nitrogen availability and organic carbon in the substrate, which could not affect photosynthetic performance. Furthermore, according to the study of Vigor, as being proved that the biochar application promotes plant growth through auxin up regulation. While in Salisbury, according to the study of Salisbury, found that a possible crosstalk between plant phytochrome photoreceptors and auxins content in promoting plant development. NPQ is one of the consequences of the fluorescent quenching phenomenon in the light, during which energy is converted to heat as being gathered from the sun, but in this study it used fluorescent. It increased the efficiency and heat dissipation occurs. While in this study, the parameter was not significantly influenced by light type or biochar application, either alone or in combination. Indeed, the different types of light spectra only slightly affect NPQ in control seedling, while no effect was detected in biochar amended ones. Although many studies have reported a positive effect of biochar application in stomatal density and conductance, in this case or in this study, the addition of biochar did not change stomatal conductance. The larger water retention capacity of biochar may cause a reduction in root water availability as being shown in the data of this study, including a water deficit and determining plant stomatal closure. However, in this journal, this was not the case since water availability was high throughout the whole experiment or the experimentation. When stomatal conductance was analyzed in response to lighting type and to the interplay between the light and biochar, no statistically significant effect was found. It has been widely reported that specific mechanism of several photoreceptors might promote stomatal opening and density depending on the wavelength of incident light. In the data, although seedlings grown with AP67 showed the highest values when biochar was added, a strong decrease of gram was measured in biochar treated seedling grown with fluorescent light spectra. Thus, in the findings, it supports the hypothesis of the study that stomatal opening is related to both spectrum composition and biochar presence. Indeed, about the study of Jensen, the relation between the stomatal activity and stomata density found an increase in in stomata density in response to increasing ratios of blue light and decrease in stomata density with decreasing ratios of green light. Likewise, it has been seen that green light exposure reversibly decreases stomatal conductance in lettuce, leading to an unfavorable effect on the plants. That was according to the study of lettuce. It including the decrease in chlorophyll content and inhibition of stomatal opening. While in this study, fluorescent light has the capacity of to lessen the extent of AP67-3L light, are characterized by the highest percentage of green light and thus negatively affects stomatal activity, especially when amended with Biochar. And now we're done to our results and discussion. And for our conclusion, the morphophysiological traits here in this journal reported represent the first attempt to unveil plant growth mechanism in response to modification in light spectrum and substrate characteristics. The present work showed that the Biochar application to a soil-less substrate has a general positive effect on above and below the ground plant morphology. In particular, Pisum sativum seedlings grown with the fluorescent control or AP67 light types doubled their fruit yield. P seedlings showed the greatest above ground and below ground plant development when a light spectrum with low red to far red ratio was used. This might be due to adaptive advantage for competition as a shade avoidance response. However, when Biochar was tested alone, it did not improve PS2 activity, but interestingly, when combined with a specific optimal light spectrum, Biochar may improve pea seedling physiological performances and change biomass organ allocation. This allows us to infer a positive synergetic effect or synergic effect of enhanced substrate characteristics. Due to Biochar application and increased plant growth and development due to AP67 light spectrum, thus it demonstrated that closed plant production systems might become more efficient by selecting an optimal LED light spectrum and including Biochar amendment in the horticultural production processes. And as a summary of this journal, light quality and nutrient availability are the primary factors that influence plant growth and development. In a context of improving indoor plant cultivation while lowering environmental impact practices, it investigated the effect of different light spectra, 3 provided by light-emitting diodes and 1 by a fluorescent lamp on the morphophysiology of Pisum sativum L. Seedlings grown in the presence and absence of Biochar. It found that all morphophysiological traits are sensitive to changes in the red to far red light ratio, with a LED type characterized by lowest red to far red ratio of -2.7 by AP67. It showed good plant development both above and below ground, especially when Biochar was presented. While Biochar alone did not affect the physiological traits, which were influenced by the interplay with lighting type, AP67 LED type had a negative impact only on the leaf fluorescence emission in the light conditions, which was further exacerbated by the addition of biochar to the growing media. However, it found that the combination of biochar with a specific optimal light spectrum may have a synergetic effect, enhancing pea seedling physiological performances and fruit yield and fostering desired traits. This is a promising strategy for indoor plant production while respecting the environment. So that's all for our journal review, fifth journal review of today. So that's all. Thank you and God bless.